Hi, we're Group 2, and the name of our presentation is Miracle Beans. I'm Adria De Apera.
I'm Tanya Alayas.
I'm Martin Estrada.
I'm Lina Aguilar.
And I'm Andrea Godinez.
My name is Tanya, and I will be introducing our project. Our experimental project includes the transformation of soybeans in order to increase their nutritional value by improving the yield of methionine. This will be done by incorporating 2 human genes into the plant. The image shown below is the soybean plant known as Glycine Max. In comparison to other plants, it is high in protein. However, soybeans cannot be considered to be high in protein overall since they have a deficiency in methionine production.
Hello, my name is Andrea, and I'll be talking about the rationale of the project. Methionine plays an important role in many cell functions. The body uses methionine to make proteins and other important compounds. In order to function properly, methionine synthase requires methylcobalamin and methionine synthase reductase, which is produced from the MTRR gene. This is why methionine is a unique amino acid, and increasing the yield of methionine works because it contains sulfur and can produce other sulfur-containing molecules in the body. It is also involved in starting protein production in your cells. Methionine can be converted into several sulfur-containing molecules with important functions, such as glutathione, taurine, SAM, and creatine. These molecules are critical for the normal function of the cells in your body. We are looking at one of the most interesting roles of methionine, being that it can transform into an SAM molecule. SAM can change your DNA by adding a methyl group, a carbon atom, and its attached hydrogen atom to DNA.
Hello, my name is Lina, and I'm going to be talking about the method of research. To create an entry clone, the idea is to create a plasmid donor, referred as P donor, using PCR and topocloning. The P donor will contain the sequence of the MTR and MTRR gene. Topocloning does not use restriction enzyme or ligase, and doesn't require post-PCR procedure. It relies on the basic ability of complementary base pairs, adenine and thymine, to hybridize and form hydrogen bonds. Using Taq polymerase during the PCR, we use topoisomerase 1 as both the restriction endonuclease and ligase by cleaving. Using the topo method to generate the P donor, we'll eliminate the use of a particular restriction enzyme during PCR. The plasmid donor will be constructed so that one gene is flanked by the ATT1 and ATT2 recognition sequences, while the other is flanked by the ATT3 and ATT4 recombination sequences, so that both genes can be transferred at the same time. The P donor will allow the recombination into the PK7, M34, GW2, 8M21, GW3G plasmid. This is a gateway plasmid designed with a PROLD promoter found in Agrobacterium rhizogens that is highly active in roots, an IOCS terminator which gives us the ATT recombination sequence that are needed for the P donor. The plasmid is part of the multi-site GainRead technology that allows for the simultaneous cloning of more than one gene. A fluorescent mark reporter that expresses GFP was used to permit visualization of the expression. The destination vector used to generate an expression clone is part of the Agrobacterium system, a gram-negative bacterium that naturally infects different plants. The use of a disarmed strain of Agrobacterium tumefaciens, a pathogenic bacterium that affects the soybean, will allow the introduction of the MTR and MTRR gene into the plant. The gateway plasmid will be grown in the Agrobacterium in vitro, and the addition of acetosergon will help to select the most viable agrobacterium to use. We are also guided by the marker that is expressed by the gateway plasmid.
Hello, my name is Martin Estrada, and I will be going over the ethics portion of our experiment. If our experiment is a success, we will have engineered a methionine-rich soybean. Our experiment is ethical by its honest means to improve nutrition. The soybean can be used to benefit both humans and animals. For the animals, the soybean will be used to incorporate a higher amount of methionine in animal feed. For humans, this can be beneficial in many ways, but most importantly, benefits people who do not incorporate meat into their diets. And it also helps babies. For the people who do not eat meat, our product would be an ideal source of obtaining nutrients without consuming meat products. Many times, people who transition to a meat-free diet get sick due to a lack of nutrition. Methionine is one of the branched amino acids that humans need to intake through their diets, since our bodies do not produce it on their own. Our soybeans will be able to improve the nutritional value and further improve soy-based diets. As for babies, baby formulas also contain soybeans. Since it is very important for babies to get as much nutrition as possible, if our soybeans are incorporated into their baby foods, our product can further help supplement infants with an alternative ingredient they can utilize. We look to further help with nutrition not only in humans but also in animals by helping companies cut back on costs as well as maximizing their nutritional value and raise healthy livestock. This is significant because embedding more amino acid supplementation into animal feed through our soybeans, we will be able to lower diet costs and maximize production. An average feeding of the two amino acids, lysine and methionine, in an average animal diet can cost approximately 20 cents per head per day. A study was done on amino acids and control feed costs, and companies can save 15 cents per pound of canola they replace. If they replace several pounds of canola in a typical diet, they can save 45 cents per head per day. Our soybeans will help them save more money by replacing the amino acids in their feed and even more on canola. Our product will also benefit countries who are starving or not getting enough nutrition due to lack of resources. With our soybean, we can introduce a low-cost, nutrient-dense option for these countries. If this is a success, we would eventually be able to get rid of the meat in our diets by eating soy-based products. This will eventually help cut out meat from our diets, and we will help the environment in the process since greenhouse gases are a major issue in our society. Thank you.
My name is Audrea De Apera, and I will be talking about the anticipated effect. The purpose of this initial experiment is to see if Argobacterium will accept the modified plasmid that contains both the MTR and the MTRR genes. We expect the acceptance of genetic modification of Argobacterium to be a success. Once we have an effective trial, we will then be able to proceed with the next phase of the experiment, which is introducing Argobacterium to the soybean plant, Glycine max. We will first introduce the modified Argobacterium to Glycine max to see if the production of methionine increases. If the methionine production levels do not change, then we will go back to the plasmid and look for another source of the gene that will be successful. If the methionine production levels increase, then we will go back to the first plasmid and insert the Krelox protein gene to remove any antibiotic resistance genes so that we may be able to release the product into the public. The overall purpose of this experiment, including all of its phases, is to increase the nutritional value of soybeans by increasing the production levels of methionine. Methionine is an essential amino acid that plays an important role for cell function. It is also one of the amino acids we use to build proteins. When the Krelox protein gene is removed, we can then introduce the genetically and nutritionally enhanced soybeans to the public. Our targeted consumers are areas in the world that lack proper nutrition and have many cases of malnutrition in their communities. It is both cheap and rich in calcium, iron, vitamin C, and many other nutrients, but most importantly, rich in protein. The modified soybeans will help to feed and sustain the people in these poverty-stricken areas and communities.
Taking everything we have talked about into consideration, our soybean will overall help in many ways, including a means to nourish people in third world countries where nutrition is at an all-time low. It will also be a step to decrease the intake of meat since our soybean will be rich in methionine, which will create a domino effect and hopefully decrease the ongoing problem of greenhouse gases in the atmosphere since livestock is known to be one of the biggest producers of methane.